You the DNA के जो these are bases. Yeah, you see this red and blue, yellow and green, red and blue. So they are the bases. We call them bases. Can you believe there are 3 billion, that means 6 billion chemicals or nucleotides in every single cell of the body, whether it is skin, brain, heart, anywhere. And how many cells in the body? Can you hear? 40 trillion. 40 trillion cells in the body. And every single body has, every cell has this DNA. Okay. So anyway, I'm going to show you chromosome. So this is divided into chromosome. The whole structure is called genome, which is divided into sections, into chromosomes. So again, to stress that this is 3 billion pairs of bases into every single cell, and that is the blueprint, or that is everything happening in your body, that determines. It has a coding. It will dictate that how many hair on your head, how your heart is going to function or work, how big, how tall you are, what color of the skin, everything is written in this. And then, once you look into this, Paramatma, there is nothing can explain. I'll give you some other examples. That how we cannot stop admiring जो भी है so Paramatma, the Supreme, who has provided this, such a designer. So amazing the things they do. So now what happens is, this is and this is just not sitting there. It is very active. It will show you in a minute, it's splitting. And after splitting, it makes a copy. And that copy is called RNA. DNA stands for deoxyribonucleic acid. And then it splits and makes RNA. This is called messenger RNA. Just see what's happening. And the messenger RNA comes out of the cell and go out and does what it's supposed to do. For example, you ate and sugar gone up. There are monitors in the body, in the blood, in the cells. They will register exactly right away. Here the sugar is high, sugar gone up. This will send here the message that yes, we need insulin. So one section of this, a gene which makes insulin, it gets into action right away. It splits, make a copy called RNA, messenger RNA, and it goes out in the cytoplasm into the cell and makes insulin. Another something really wonderful I want to share with you, that now for the scientists, we have discovered the structure of the insulin and the structure of the gene which makes insulin. Okay? Gene is just a portion of the DNA which has a very specific function depending on the specific sequence. Now this again you have to understand. These bars, only 4 letters. A, T, G, C. That's it. And that language to make every part in your body. That language tells you how to make heart, how to make your brain, how to make your muscles, how to make your bone. Red blood cells. Oh my God. Can anybody guess how many red blood cells body makes every day?
Billions.
Or every hour? Any guess?
Billions.
Yeah, somebody knows. 2.5 million. You know how often? Every second. When I read this first time, I keep going back to the book. I said, really? and I will forget every second, you know. On one hand, we talk about this earth moving around the sun, and again there is speed, you know how, 67,000 miles an hour. That's one wonder, you know. And there's in the cell 2.5 million every second. Can you imagine any manufacturing plant? And red blood cell is so complicated. Quaternary, you know, protein which gets the oxygen from the lungs and take it to the tissues. It's not just something simple. So this is what, you know, it does. So anyway, so that section of the DNA which makes insulin, we have identified that this sequence is A, T, G, C, T, T, G, whatever sequence. And those nucleotides of the chemical, they are put together, the scientists, in the lab. Okay? So for example, here in the lab, in the dish there, we put this sequence together. We made the gene which can produce insulin. But nothing happens in the lab. Nothing. As soon as you pick it up, put it into a living cell, whether the human cell, plant cell, or animal cell, you start producing, you know, bacteria. It start producing insulin. So what is that thing? Something living in that cell, which makes it to work, function, you know. So I don't know if you know that the insulin we call human insulin. I'm sure a lot of people know. So that insulin, that's how it's made. It is made like this. They have made this gene in the lab and put it into the cell of bacteria, and then bacteria, of course, they will grow like anything. So they put in the petri dish, and by the morning you have all kinds of, you know, bacteria, and then they extract the insulin. Just to give you one example, it's just one example. There are millions and millions, actually billions, of reactions going on in the body every second. You know. So it's just amazing how these 4 letters, A stands for adenine, is attached to T, is tyrosine, then C is cytosine, and G is guanine. That's it. These 4 chemicals, and they have their own language to make whatever we have in the body. So what it does, that's another miracle here. These are the amino acids. In the body, we have about 20 amino acids. 12 amino acids we can make ourselves, 8 we have to take from outside. So this instructions from this DNA, or the part of DNA gene, it will instruct which amino acid and the sequence to make this. And the other part of this will tell how to fold it. That folding makes all the difference. Even if you make this structure but you fold wrong, it won't function. And this is happening. You know, I mean, insulin is just one section. There is a disease, and then if any mutation, there is a disease called phenylketonuria. It's a defect. The child is born with that, and the slide there I will show you. The sequence has to be CCG, and normally in that person is CGT. That's it. Just one letter missing, a different, you know. Instead of G, it is a T, and there is no further reaction. So that collects in the blood and causes havoc. The children who have this phenylketonuria, they are retarded mentally, they don't develop, and their life is nearly 30 to 40 years. Just one letter. Of course, now we know how to correct it, and things are advancing. So that just gives you something to think about, that how magnificent and how vast, you know, Vishalta in the cell, the structure and the function, you know. Now comes to the real part, the health. How it works. Anyway, I'll show you. There are some slides there where you see a blood vessel, nice and smooth. Okay? And with our food and the lifestyle, you start to get the deposits inside the wall. So anyway,
जायज की लाइट में कम कर देना स्टेज की कम करो स्टेज की
स्टेज लाइट कम
स्टेज लाइट
ये ऊपर लाइट कम
स्टेज लाइट सब बंद कर दो सब बंद कर दो लाइट ये चेयर भी इधर कर दो
ऊपर की लाइट बंद कर देता
लाइट बंद हो रहा है दीपक कहाँ है
भारत के सबसे ज्यादा
तो देखिए कोई नहीं है
so there's the DNA, and okay, go back to the DNA. Yes. The next slide.
दीपक लाइटें बंद करो कुर्सी हटाओ
See how it is splitting. You can see how the DNA is splitting. This is happening inside the cell. And that the little circle you see, that's a monitor. It will tell you how much to split and what function.
बंद करो साइड बंद करो ये भी बंद कर दो वो भी बंद कर दे
ये पहले बंद करेगी थी ना ये भी बंद कर दो
ठीक है ना?
ये चेयर भी थोड़ी साइड
और ये अब साफ हो गया
Okay, next.
वो enlarge
Yes. That's again showing that how they are splitting and making RNA, and that RNA goes out of the cell and takes instruction. Okay, next.
ये लाइट भी बंद कर दो बेटा
ये लाइट भी बंद कर
Next.
दो
हाँ. Okay.
दीपक ये लाइट भी बंद कर दो बेटा
साइड पे करो सारी लाइट्स बंद कर दो ऊपर की
ये वाली लाइट बंद कर
Okay, look at this one. You see the one on the left side is a normal. Okay. One on the right side, you see those spots? These are the spots. There is the damage, and they can again create problems. And what causes the damage, which I'll come back to that, let you know. So this damage will make you sick. That's the reason. Okay, next. Yeah, anyway, okay. Alright. The one before that was just showing you that amino acid chain where the CCG becomes CCT and then you have a problem. So, but anyway, this one is the one I was going to talk. This is blood vessel. The life depends on the smoothness of the blood vessels in the whole family, whole body. Okay?
उनको माइक कैसा दे दो हाथ में पकड़ा दो
Okay. Okay. Good. Yeah. So look at the left, all smooth and nice, and now you see this little plug. Okay? This plug again, cholesterol and some fat and some calcium and all that, you know. So by genome sequencing, you can predict about these diseases. Diabetes, hypertension, atherosclerosis, heart disease. This plug, if it's in the artery, which is going to the heart, and next slide. See, it becomes bigger. Then next, and now the whole thing is clotted. So there is no blood going through, and that's how you get the heart attack and the stroke. Wherever. If there's artery in the brain, you got a stroke. If there's artery blocked in the heart, you get a heart attack. If the artery is in the leg, you get a gangrene, you know. So this is what happens, and this is preventable. The good news is that this is preventable. There are still in US, there are 500,000 heart attacks. People dying with that. 500,000 every year. And so many, they have dropped. And because they have never been checked, they never take care. And this is what happens. Okay, next. This is again showing that how the things are progressing. Suppose this is a defective gene there. So one can go, it's not permanent right now. But a day will come where you can go in there and take this gene out and put a correct one. Now one thing I tell you that in the body, when there is a replication, of course 3 billion bases, you know, it's bound to have some mistakes. So they are inspectors, literally inspectors, their protein going up and down. Their function is to identify if it is ATGC or not. If it's ATGG, they go right there and there's a repair enzymes and they fix it. What happens when you get older? And of course we try to get, you know, stay young. So what happens is that there are too many mistakes. And those inspectors and the monitors and the repairmen, they all get slow as well. And that's why all these mutations, they collect. And you get the diseases, heart disease or dementia or cancer. Same. So, okay, next. Now this one I want to show you that never give up. This start on the top is the section to the leg, to the thigh. All the red is the muscle, in the center is the bone, and outside there's a little trim of fat. Look at this slide below. Very little muscle, somebody, on the top somebody 40 years old. The bottom somebody 80 years old. Okay? But then there's a guy who started working out at 70. And look at below, this is about 70-year-old guy. Back to the muscle. So you can regenerate if you have a proper lifestyle and exercise. You can become younger again. This is proof and it is documented. Okay, next. Let me finish. Okay, next. Yeah. Same. This is just to show that this one. DNA go to RNA, and the RNA takes the information and makes this chain of amino acids, and the chain of amino acids is called proteins. And proteins can be for the structure of the body, could be for the enzymes, could be promoters, could be inspectors, all different functions. That's what. So these two things, DNA, the instructions, and this amino acid in the whole body, in every single cell. Okay, next. Yeah, this is this called epigenetics. So that's what I come to when I finish this. Epigenetics is the one. It has proteins, it has certain other chemicals, it has some vitamins, some other nutrients. They surround the DNA, and they can affect the function of the DNA, of the gene. Okay. So gene or the DNA is not the destiny. It can be modified with the lifestyle, proper diet, even meditation, good sleep. So they can modify the expression of the gene. Okay, next. Yeah, this is just the, this part I was showing you, ATGC. This is the structure. And they are here. But if you open that up, this is what they look like. There's just a bunch of chemicals. And that's it in the DNA. Okay, next. And then, they are just showing this is the chromosome, which is packed with the genome. And then the video. Now I want to show you a video to show you that how we do the sequencing now. We take a sample, either the saliva or the blood, and I'm outsourcing. There's a video there. Shall I show you?
नहीं मैं तो कहीं आई हूँ लेक्चर हो रहा है
Yeah. So just try to listen to this one.
Genomic sequencing is a process where the analyzing a sample of DNA taken from your blood. the lab,
कोई आया हुआ
technicians
है
extract DNA and prepare it for sequencing. Within every normal cell are 23 pairs of
मैं
chromosomes.
घर आऊंगी ना तो
Chromosomes are structures that house DNA. DNA is coiled into a shape called the double helix. The double helix can be unwound into a ladder shape. The ladder is made up of paired chemical letters called bases. Altogether, our DNA contains about 6 billion such paired chemical bases. There are 4 bases in the DNA alphabet: A, T, C, and G. In DNA, base A joins only with base T, and base G joins only with base C. To read the sequence of bases in DNA, samples are inserted into a sequencing instrument, where high-frequency sound waves break the DNA into smaller pieces that are only about 600 bases long. Special tags are added to the ends of the fragmented DNA. These tagged strands of DNA can then attach to a glass slide. In a sequencer, each piece of DNA is copied hundreds of thousands of times, which creates clusters of identical DNA fragments. Next, the sequencer reads the DNA one base at a time, using different colored tags for each DNA base. Special sensors within the machine detect the different colored tags. This sequence of colors reveals the DNA sequence of each fragment. Powerful computers piece together these individual DNA fragments and reveal the sequence of
Okay,
DNA.
you can stop it. See that, okay, the leave this. You know this sequence, this was sequencing. You sequence the DNA and this report comes out. So the A, G, G, C, 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 G,
आपकी
G.
आवाज नहीं आ रही.
This sequence, you saw this, the lab have this sequencing done of the DNA. And these letter sequence will come in the form of a report. Then they have collected data, millions of data, points. And that pattern of the sequence will predict that what problem is going to have when you're 40 or 50 or 60 or 70. Okay? So, for example, I was showing you that art is the plaque. So, if you have a sequence which is indicative of somebody going to have a plaque and an artery of the heart, then we can work on it. We can try to change the expression of the gene. So now, let's come to what we can do to prolong life and live healthy. There are so many. There's Dr. David Sinclair at MIT, which I just met him a couple of months ago. He, of course, claimed that 150 not a big deal, one can do that. But let's tell you this, that the best and the cheapest and proven fact is that when you eat and what you eat. So the most important now they come up that we eat just too much, too often. Every time we eat, the insulin goes up. And insulin going up will cause damage to the lining of the artery. So now the recommendation is eat in a narrow window of 6 to 8 hours. Say for example you eat at 6 o'clock, in the evening at 4. Nothing till morning, about 9 or 10. That's how it's either breakfast and then lunch and dinner together. That's it. And nothing in between. Nothing. That is the best. If you practice that, you will see the difference within 3 weeks. Within 3 weeks, your weight or your stomach, you'll feel more energetic. You'll sleep better. Your digestion will get better. So all those things are such a simple thing. But again, not easy to do it, I understand. But once you know what you're doing, whenever we eat extra, all those calories are already so busy, 3 billion. And then you clog these cells with these extra calories, the fat and the triglycerides and everything else. So just imagine how hard for the cell to function. And that's how we get this damage, all the spots, damage of the DNA, So that's number one. Number two is avoid anything processed, synthetic, All these burgers and fries and everything else, fast food, very, very dangerous. Acrylamide, so dangerous. Plant-based diet. This is the picture to show, to impress. that if you eat greens, fruits and vegetables, this is how you're going to look like. And if you eat burgers and fries and coke, that's what is going to happen. So this is how you get the damage to the DNA, and then you get obesity, then you get heart disease, and then you get a stroke, and that's it. So it's not out of reach. Even today, if you decide, hey, I have to live healthy, I have to live long, and I want to be healthy, you can start. Just eat twice in a narrow window of maybe 6 to 8 hours. 8 hours is okay, not too bad. A lot of patients might have, they will eat their breakfast at 8 and the last meal at 4 o'clock in the evening. So 16-hour fasting. You can do 17, 18, more the better. 19 is the best ideal. That's what the research has shown. And stay on the fresh, as much as possible, plant-based diet, fresh. So that's the next best thing. And then what I recommend to my patients, we have a basic lab and certain things which we know we need to do, and we give certain supplements. Couple of things. One is vitamin D. I find in my patients, many patients deficient, so they need vitamin D. Vitamin D is so important. In fact, during COVID-19, those who were really sick, most of them, they were deficient in vitamin D and otherwise. Then magnesium. This Dr. David Sinclair, he prescribed some called resveratrol, which is out of the grapes, skin, and there's another chemical called NMN, which keeps the DNA healthy. So there are certain chemicals, of course. I'm not prescribing anything, but they are. That's what he is using. So, but again, something simple. Go on a diet which I'm telling you. Eat only twice a day, and narrow window of maybe 6 to 8 hours. That's it. And as much as possible, plant-based diet, and that itself will make a big difference. You can see within a month, absolutely, and you can thank me. So with that, I think I'll close. Thank you again for your attention. I know it may be going over your head, something, but I try to make it simple, and I hope you understood. Any open for questions, if anybody has? Yeah.
I have a question.
Okay.
May I request you, please? It would be better. We are all oldies, sitting in court in order to get something from you. And this content is very good for doctors, I do understand. But let it be something, few tips you can give us, which we can take home with us, so that we can apply them in our daily life.
That's the one.
Yeah.
That's the one I told in the last.
Yeah,
Yeah.
please.
Just do. Forget about supplements and just
Note
eat.
them down and go back with them
Yeah.
and try
Okay.
them. Please.
थैंक यू सो मच सर। मुझे ऐसा लगता है कि हम डॉक्टर्स को तो ये डीएनए और आरएनए की भाषा बहुत अच्छे से समझ में आती है, बट जनरल हम कुछ टिप्स शायद सर से एक्सपेक्ट कर रहे हैं कि कुछ ऐसे टिप्स दिए जाएं ताकि हम अपनी एज को, डेफिनेटली डीएनए डिसाइड करता है, बट सर ने जैसे बताया तो मैं थोड़ा सा शॉर्ट में बताती हूँ जितना मुझे समझ में आ पाया कि डीएनए हमारी एज तो डिसाइड करता ही है डेफिनेटली, बट कुछ चीज़ें हमारे हाथ में हैं जिससे हम अपनी एजिंग प्रोसेस को रोक सकते हैं वहीं पर। एजिंग इज नॉट अ डिजीज, इट इज डेफिनेटली नॉट अ डिजीज, इट इज अ प्रोसेस। हम सबको इसको पार करना है, हर स्टेज को क्रॉस करते करते हमें एक एज पे आके स्टेबलाइज होने का मन करता है कि अब ये एज नहीं रुक जाए। उसके लिए डॉक्टर साहब ने जो लास्ट में बताए कुछ टिप्स शायद हमने मिस किए भी हैं या शायद हम नहीं गेन कर पाए एकदम से। लाइफस्टाइल, आजकल मैक्सिमम प्रॉब्लम्स जो है वो लाइफस्टाइल डिसऑर्डर्स से हो रही हैं। वो लाइफस्टाइल को अगर हम थोड़ा सा चेंज करेंगे, बर्गर्स नहीं खाएंगे, फ्राइज नहीं खाएंगे और ये सब चीज़ें हम थोड़ा सा अपने लाइफ में चेंजेस लाएंगे तो डेफिनेटली एक अच्छी हेल्दी लाइफ बिता सकते हैं। अपनी एज को रिस्पेक्ट देते हुए जो चीज़ें हमें लगता है कि हमें नुकसान दे रही हैं उनको हम रोक सकते हैं। सो थैंक यू सो मच सर। अभी हमारे बीच में एडवोकेट नेहा चौधरी जी हैं जिनको हम हीलिंग क्लब की तरफ से फैसिलिटेट करना चाहेंगे। कंचन मैं प्लीज़ आपको